Hey everybody, welcome back to Chapter 3, Biomolecules. Before we get started with the lecture today, I'd like for you to check out this video, and then whenever you get done with that, come back to this current video, and we will get started talking about biomolecules. So every organism needs energy in order to survive, and how we get energy is by eating food. Well, within food are critical nutrients that our body needs to survive. A lot of these critical nutrients that we need are what we call biological macromolecules, or for short, biomolecules. There are 4 types of biological macromolecule classes. First being carbohydrates, the second lipids, third proteins, and fourth nucleic acids. Each of these 4 major biological macromolecule classes is an important cell component and provides a wide array of functions. Biomolecules are organic, meaning they contain carbon. They will also commonly contain hydrogen, oxygen, and nitrogen, as well as some other minor elements too. The formation of these macromolecules requires the combining of subunits called monomers. Monomers will join together through covalent bonds to form larger molecules known as polymers. When this happens, monomers release water molecules as byproducts. So this type of reaction is called dehydration synthesis, which literally means to put together while losing water. Here's an example of dehydration synthesis. This is the synthesis of maltose. 2 glucose molecules covalently bond to form maltose and a byproduct of water. The opposite of the dehydration synthesis reaction is hydrolysis. In hydrolysis, polymers break down into monomers. This happens through a chemical reaction in which the polymer breaks down into 2 components. One part gains a hydrogen atom and the other gains a hydroxyl molecule. Both the dehydration and hydrolysis reactions can be sped up by enzymes. Catalytic enzymes in the digestive system hydrolyze the food we ingest into smaller molecules. This allows cells in our body to easily absorb nutrients in the intestine. There's a specific enzyme that breaks down each type of macromolecule. For example, amylase, sucrase, lactase, or maltase are examples of enzymes that can break down carbohydrates. Other enzymes called proteases, such as pepsin or peptidase, can break down proteins, and lipases can break down lipids. This reaction shows the hydrolysis of maltose, which is the opposite of the dehydration of maltose. In this reaction, water is added to maltose, creating a reaction that produces 2 glucose molecules as a product. The first class of biomolecule we're going to cover are carbohydrates. Carbohydrates exist in a carbon to hydrogen to oxygen ratio of 1 to 2 to 1. This formula explains the origin of the term carbohydrate. The components are carbon, carbo, and the components of water, hence hydrate. One of the main purposes for carbohydrates is for energy storage. Cells store carbohydrates in the form of sugar. Sugar can then be utilized later to perform all types of metabolic processes. Scientists will classify carbohydrates into 3 subtypes: monosaccharides, disaccharides, and polysaccharides. Monosaccharides are also known as simple sugars, the most common of which is glucose. Monosaccharides can be named by the number of carbons they have, which usually ranges from 3 to 7. If the monosaccharide has 3 carbons, it's called a triose. If it has 4 carbons, it's called a tetrose. If it has 5 carbons, it's called a pentose, and 6 carbons would be called a hexose. All monosaccharides will end in the suffix -ose. So it's an easy way to determine whether or not the molecule you're looking at is a monosaccharide. We can also classify monosaccharides based on the position of their carbonyl group. Aldoses have a carbonyl group at the end of the carbon chain, and ketoses have a carbonyl group in the middle of the carbon chain. This example over here on the left that I'm circling shows an aldose because its carbonyl group is located at the end of the carbon chain. This right example shows a ketose because its carbonyl group is located in the middle of the carbon chain. Now, the second set of examples shows how the number of carbons can affect the carbon chain backbone. So on the left over here, we have 3 carbons, so it's a triose. In the middle, we have 5 carbons, so it's a pentose. And on the right, we have our most common monosaccharide, glucose, which is a hexose and has 6 carbons. 3 monosaccharides that are important to know are glucose, galactose, and fructose. They're all isomers, meaning that they have the same chemical formula, but a different arrangement of their atoms. Glucose is an important source of energy for humans. During cellular respiration, energy releases from glucose, and that energy helps to make ATP. Plants synthesize glucose using carbon dioxide and water, and glucose in turn provides energy requirements for the plants. Humans and other types of animals that feed on plants often store glucose as catabolized starch. Galactose is found in milk. It's part of lactose, or milk sugar. Fructose is found in sucrose and is the type of sugar found in fruit. Monosaccharides can exist in a linear chain or as a ring-shaped molecule. In aqueous solutions, they are typically found in ring forms. 5 and 6 carbon monosaccharides exist in an equilibrium between their linear and ring forms. Glucose in a ring form can have 2 different hydroxyl group arrangements around the anomeric carbon. The anomeric carbon is the carbon 1 that becomes asymmetric in the ring formation process. If the hydroxyl group is below carbon number 1 in the sugar, it is considered alpha. If it is above the plane, it is considered to be in the beta position. So if you'll take a look at this image, on the left we have glucose. This glucose molecule is in the process of forming a ring. Now it can position the hydroxyl group in 2 different locations. It can position it below the ring plane, which is said to be in the alpha position, or it can position it above the ring plane, which is said to be the beta position. This image is just illustrating for you that fructose and ribose can also form rings. However, they form 5-membered rings because they have a 5-carbon backbone instead of the 6. The next type of carbohydrates we're going to cover are called disaccharides. Disaccharides literally means 2 sugars. So it's a monomer composed of 2 monosaccharides joined together. During this process, one monosaccharide's hydroxyl group combines with another monosaccharide's hydrogen, which releases a water molecule and forms a covalent bond. We call this type of bond a glycosidic bond. Glycosidic bonds can also be alpha or beta, depending on whether the oxygen ends up above or below the ring plane. Common disaccharides include lactose, maltose, and sucrose. Lactose, which is naturally found in milk, is a disaccharide consisting of a glucose and galactose molecule. Maltose, or malt sugar, is a disaccharide that is formed when 2 glucose molecules have covalently bonded together. Maltose is often found in wheat beers. The most common disaccharide is sucrose, or table sugar, which is composed of glucose and fructose monomers. The last type of monosaccharide we're going to cover is called a polysaccharide. Polysaccharides consist of long chains of monosaccharides linked together by glycosidic bonds. These chains may be branched or unbranched, and it may contain different types of monosaccharides. Starch, glycogen, cellulose, and chitin are 4 common types of polysaccharides. Starch is the storage polysaccharide found in plants. Plants store starch in the form of sugars. An amylose and amylopectin mixture comprise these sugars. Plants are able to synthesize glucose, and they store the excess glucose as starch in different plant parts, including the roots and seeds. Humans consume many types of starches. An example of this would be potatoes. In order for humans to break down that starch that they consume, enzymes are necessary. For example, amylase is present in saliva catalases, which breaks down the starch into smaller molecules such as maltose and glucose. The cells can then absorb the glucose. Glycogen is the animal equivalent of starch, or in other words, it is the storage polysaccharide in animals. It is a highly branched molecule usually stored in liver and muscle cells of humans and other vertebrates. It is composed of multiple monomers of glucose. Whenever blood glucose levels decrease, glycogen breaks down to release glucose. Cellulose is the structural polysaccharide found in plants. It is the most abundant natural biopolymer. Cellulose is mostly found in a plant's cell wall. It provides the cell structural support. Wood and paper are both mostly cellulosic in nature. Cellulose is so important to plant cells because they provide the cells with rigidity and high tensile strength. This is because every other glucose monomer in cellulose is flipped over, which allows the monomers to be packed extremely tightly as they extend in very long chains. Human digestive enzymes cannot even break down the linkages that are formed within the cellulose bonds. Herbivores such as cows, koalas, and buffaloes are able to, though, with the help of specialized flora in their stomach. Some of these animals have specific species of bacteria and protists in their rumen and secrete the enzyme cellulase. The appendix of grazing animals also contains bacteria that digest cellulose. Another type of structural polysaccharide is chitin. Chitin is found in the exoskeleton of arthropods. Arthropods have an outer skeleton called the exoskeleton, which protects their internal organs. The exoskeleton is made out of a polysaccharide containing nitrogen called chitin. It is made of repeating molecules, which are a type of modified sugar. Chitin can also be found as a major component of fungal cell walls. Lipids are a diverse group of molecules that consist of largely nonpolar compounds in nature. This is because they are composed of hydrocarbons that include mostly nonpolar carbon-to-carbon or carbon-to-hydrogen bonds. Now remember, nonpolar means that a substance is hydrophobic or insoluble in water. Lipids can perform many different functions within a cell. They are capable of storing energy for long-term use, providing insulation from the environment for plants and animals. They help keep aquatic birds and mammals dry by forming a protective layer over fur or feathers because they are water repellent in nature. They also form the building blocks of many hormones and are an important constituent of all cellular membranes. Lipids include fats, oils, waxes, phospholipids, and steroids. A fat molecule consists of 2 main components, glycerol and fatty acids. Glycerol is an organic compound with 3 carbons, 5 hydrogens, and 3 hydroxyl groups. Fatty acids have a long chain of hydrocarbons to which a carboxyl group is attached. The number of carbons can range anywhere from 4 to 36. The most common are those containing 12 to 18 carbons. The fatty acids attach to each of the glycerol molecules 3 carbons with an ester bond through an oxygen atom. Because fats have 3 fatty acids attached to a glycerol, they are called triglycerides or tricyclycerols. Fatty acids may be saturated or unsaturated, however they all end with a carboxyl group. Whether or not the fatty acid is saturated or unsaturated depends on whether the hydrocarbon chain contains a double bond or not. An unsaturated fatty acid will have a double bond located within the hydrocarbon chains. Most unsaturated fats are a liquid at room temperature. We call these oils. If there is only one double bond in the molecule, then it is said to be a monosaturated fat. An example of this would be olive oil. If there is more than one double bond, then it is said to be a polyunsaturated fat. An example of this would be canola oil. When a fatty acid has no double bonds, it is a saturated fatty acid because it is not possible to add any more hydrogens to the chain of carbon atoms. Saturated fatty acids typically remain a solid at room temperature because the long straight fatty acid strains. Saturated fatty acids typically remain a solid at room temperature because their long straight fatty acids with single bonds allow them to pack tightly together. A common example of a saturated fatty acid would be stearic acid, which is found in animal fats. Unsaturated fats or oils are usually of plant origin and contain either a cis unsaturated fatty acid or a trans unsaturated fatty acid. Cis and trans indicate the different types of configuration of the molecule around the double bond. If hydrogens are present in the same plane, it is a cis fat. If the hydrogen atoms are on 2 different planes, it is called a trans fat. The cis double bond will cause a kink or a bend that prevents the fatty acids from packing tightly, which keeps them liquid at room temperature. Olive oil, corn oil, canola oil, and cod liver oil are all examples of unsaturated cis fats. Now, these lower 2 unsaturated fatty acids show the difference between a cis and a trans configuration. On the top, the cis configuration shows both hydrogen atoms to be on the same plane. The trans shows the different hydrogen atoms on different sides of the plane. So saturated fatty acids, as we covered, are long chains of single carbon-to-carbon bonds in which the molecules fit relatively close together. They also have really high melting points that keep them solid at room temperature. Unsaturated fatty acids have one or more double carbon-to-carbon bonds. They do not pack closely, and they usually remain a liquid at room temperature. Trans fats are the types of unsaturated fatty acids that contain hydrogen atoms on opposite planes surrounding the carbon-to-carbon double bond. Recent studies have shown that an increase in trans fats in the human diet may lead to higher levels of low-density lipoproteins, or bad cholesterol, which can lead to plaque deposition in the arteries and result in heart disease. Margarine, some types of peanut butter, and shortening are examples of artificially hydrogenated trans fats. The food industry artificially hydrogenates oils to make them a semi-solid and of a consistency that's desirable for many processed food products. What they do is they bubble hydrogen gas through the oils to solidify them. During this hydrogenation process, double bonds of the cis conformation in the hydrocarbon chain may convert to double bonds in the trans configuration. Omega fatty acids are a type of an essential fatty acid. Essential fatty acids are those that the human body requires but cannot synthesize, meaning that they must be supplemented in our diet. There are only two types of omega fatty acids that humans must worry about: omega-3 fatty acids and omega-6 fatty acids, both of which are polysaturated fatty acids. Salmon, trout, and tuna are all good sources of omega-3 fatty acids. The consumption of omega-3 fatty acids can reduce the risk of sudden death from heart attacks, low triglycerides in the blood, decrease blood pressure, and prevent thrombosis by inhibiting blood clotting. They can also reduce inflammation and may help lower the risk of some cancers. Waxes are another type of lipid. Waxes are hydrophobic in nature, which prevents water from sticking to any type of surface that has a wax on it. Bird feathers and some types of plant leaves utilize these types of waxes to shed water. Waxes are composed of long fatty acid chains that have been esterified to long chains of alcohols. Phospholipids are a major component that comprise all types of biological membranes. Like fats, they are composed of fatty acid chains attached to a glycerol backbone. However, instead of 3 fatty acids attached as triglycerides, there are 2 fatty acids that form a diglycerol, and then a modified phosphate group occupies the glycerol backbone's 3rd carbon. A phospholipid is an amphipathic molecule, meaning that it has a hydrophobic and a hydrophilic portion. A phospholipid is an amphipathic molecule, meaning that it has a hydrophobic and a hydrophilic portion. The fatty acid chains are hydrophobic and nonpolar, meaning they cannot interact with water, and thus they make up the hydrophobic portion of the phospholipid. The phosphate-containing group, or the polar head, is hydrophilic and can interact readily with water. Here's a little better look of what the structure of a phospholipid looks like. We have a phosphate head attached to a glycerol, which is also attached to 2 fatty acid tails. This is the hydrophobic portion of the phospholipid, and this is the hydrophilic portion of the phospholipid. Now, the hydrophobic tails of a phospholipid may contain either an unsaturated fatty acid or a saturated fatty acid. Okay, now I'm going to back up a slide to show y'all something called a micelle. Now, a micelle is this little circle right here. This occurs when you drop a bunch of phospholipids into a container of water. The phospholipids will orient themselves so that their hydrophilic head will face the water surrounding them, and their hydrophobic tails will become the inside of this circle, so that it is, so to say, segregated from the water. Now, in a membrane, a bilayer of phospholipids will form because the phospholipids' fatty acid tails will face it inside, away from the water, whereas the phosphate group will face the outside, towards the aqueous side. The last type of lipid we're going to cover is called a steroid. Unlike the phospholipids and fats, steroids have a fused ring structure. And although they do not resemble the other lipids, we group them in with the other lipids because they are also hydrophobic and insoluble in water. All steroids will have 4 linked carbon rings, and several of them, like cholesterol, will have a short tail. Cholesterol is the most common steroid. The liver synthesizes cholesterol, and it is the precursor to many steroid hormones such as testosterone and estradiol. It is also the precursor to vitamin D. Each living cell may contain thousands of proteins, each with a unique function. Proteins are one of the most abundant organic molecules in living systems, and they have the most diverse range of functions of all the macromolecules. Proteins may be structural, regulatory, contractile, or protective. They may serve in transport, storage, or membranes. They may even be toxins or enzymes. Proteins can be enzymes, which catalyze biochemical reactions like digestion, or they can be hormones, which are used for chemical signaling. Proteins, which are enzymes, are usually complex proteins. Each enzyme is specific for the substrate upon which it acts. The enzyme may help in breakdown, rearrangement, or synthesis reactions. Enzymes that break down their substrates are called catabolic enzymes. Those that build more complex molecules from their substrates are called anabolic enzymes. And then enzymes that affect the rate of a reaction are called catalytic enzymes. All enzymes increase the rate of reaction and are therefore called organic catalysts. And we just learned that there were hormones called steroids. Well, there are also hormones that are proteins. Both are chemical signaling molecules. Proteins that are hormones are usually small and secreted by the endocrine cells that act to control or regulate specific processes. This table shows the primary types and functions of proteins. First, we have digestive enzyme proteins. These proteins help by catabolizing nutrients and breaking them into smaller units. Transport proteins are able to carry substances either through blood or lymph throughout the entire body. Structural proteins are able to construct different types of structures, like the cytoskeleton of a cell. Hormones can coordinate different body systems activities. Defense proteins are able to protect the body from foreign pathogens. Contractile proteins are involved in muscle contraction, and storage proteins are able to provide energy for use at later times. A few more examples of proteins include actin and myosin, which is involved in muscle contraction, insulin, which is a hormone that regulates blood glucose levels, immunoglobulin, which is an antibody, hemoglobin, which is involved in oxygen transportation, rubisco is able to fixate atmospheric carbon dioxide, ferritin is involved in iron storage, and spider silk is a liquid crystalline protein used to create webs. Rhodopsin is a light-sensitive receptor protein. All of these have vastly different structures and functions. Actin and myosin work really uniquely, and they're really neat to watch in this video. So go ahead and check this video out and then come back. Amino acids are the monomers that comprise proteins. They get their name amino acid because they contain both an amino group and a carboxylic acid group in their basic structure. There are about 20 common amino acids present in proteins. 9 of these are essential amino acids in humans because the body cannot produce them and we have to obtain them from our diet. Each amino acid has the same fundamental structure. They all contain an alpha carbon bonded to an amino group, a carboxyl group, and to a hydrogen atom. Every amino acid also has another atom or group of atoms bonded to a central atom known as the R group. The R group is what controls whether the amino acid will be acidic or basic in nature, and whether it'll be polar or nonpolar. This image shows the 20 common amino acids. You can see that the structure stays the same with all of them, except they all have a different R group attached. The protein's shape, size, and function are ultimately determined by the sequence and number of amino acids. The formation of peptides begins with a covalent bond or a peptide bond, which attaches to each amino acid and forms a dehydration reaction. One amino acid's carboxyl group and the incoming amino acids group combine and release a water molecule. The resulting bond is known as the peptide bond. products that share peptide bonds are called peptides. As more amino acids are joined to the chain, the resulting chain is called a polypeptide. Each polypeptide will have a free amino group at one end, and the other end will have a free carboxyl group. The amino acid's unique sequence in a polypeptide chain is known as its primary structure. For example, the amino acid sequences in the A and B chains shown are unique to insulin. The gene encoding for a protein ultimately determines the unique sequence for every protein. A change in a nucleotide sequence of gene's coding region may lead to adding a different amino acid to the growing polypeptide chain, which causes a change in protein structure and function. In sickle cell anemia, the hemoglobin beta chain has a single amino acid substitution, which causes a change in protein structure and function. Specifically, the valine in the beta chain substitutes the amino acid glutamic. This causes a structural difference between a normal hemoglobin molecule and a sickle cell molecule. This structural difference causes a dramatic decrease in life expectancy. Because of this change in one out of the 600 amino acids in the chain, hemoglobin molecules form long fibers that distort the biconcave or disc-shaped red blood cells and causes them to assume a crescent or sickle shape. This shape clogs blood vessels. This can lead to a myriad of serious health problems such as breathlessness, dizziness, headaches, and abdominal pain. The secondary structure of a protein is formed by the local folding of the polypeptide in some regions. The most common folding structures are called the alpha helix and the beta pleated sheet. Both structures are held in shape by hydrogen bonds. The hydrogen bonds form between the oxygen atom in one carbonyl group and one amino acid and another amino acid that is 4 amino acids further along the chain. In the alpha helix chain, the polypeptide's R groups protrude out. In the beta pleated sheet, hydrogen bonding between atoms on the polypeptide chain's backbone form the pleats. The R groups are attached to carbons and extend above and below the pleats folds. The pleated segments align parallel or antiparallel to each other, and hydrogen bonds form between the particularly positive nitrogen atom in one of the amino groups and the particularly negative oxygen atom in the peptide's backbone. The tertiary structure of a protein is based on the polypeptide's unique three-dimensional structure. This structure is in part due to chemical interactions at work on the polypeptide chain. Primarily, the interactions among R groups create the protein's complex three-dimensional tertiary structure. Then the nature of these R groups and the amino acids involved can counteract forming the hydrogen bonds that we described for the standard secondary structures. For example, R groups with like charges will repel each other, and those with unlike charges will be attracted to each other. These interactions, weak or strong, will determine the protein's final three-dimensional shape. In nature, some proteins will form several polypeptides or subunits, and the interaction of these subunits will form the quaternary structure. Weak interactions between these subunits help to stabilize the overall structure. As you've learned, each protein has its own unique sequence and shape that chemical interactions hold together. If the protein were to be subject to changes in either the pH, the temperature, or the ionic concentration of the solution, then the protein structure may change and lose its shape. Now, this loss of shape can occur without altering the primary sequence, which is what we call denaturation. Denaturation is often reversible because the polypeptide's primary structure is conserved in the process. If the denaturing agent is removed, then the protein will resume its function. Sometimes denaturation can be irreversible and lead to permanent loss of function. An example of irreversible protein denaturation is frying an egg. When you fry an egg, the albumin protein in the liquid egg white denatures when it's placed in the hot pan. To learn a little bit more about the structure of a protein, go ahead and check this video out right here. The last type of biomolecule we're going to cover is called a nucleic acid. Nucleic acids are the most important macromolecule for the continuity of life. They carry the cell's genetic information and the instructions for how it functions. The two main types of nucleic acids are deoxyribonucleic acid, or DNA, and ribonucleic acid, RNA. DNA is housed in the nucleus of the cell, and it contains all the genetic information for that organism. DNA, which is housed in the cell's nucleus and holds all of the organism's genetic information, will then be transcribed into RNA, which can then be sent outside the cell or used within the cell to translate into proteins. RNA and DNA are very similar. However, they have a few big differences as well. DNA is located inside the nucleus, whereas RNA is located outside of the nucleus. DNA contains all of the genetic information of the organism. RNA contains a segment of a copy of DNA, which then can communicate with cells. DNA controls cellular activities by activating or deactivating genes. RNA is involved in protein synthesis and regulation. DNA will be composed of cytosine, guanine, adenine, and thymine. RNA, on the other hand, will be composed of cytosine, guanine, adenine, and uracil. These are what we call nitrogenous bases. They're very important components of nucleotides. They are organic molecules that are composed of carbon and nitrogen. Nucleic acids are made up of repeating subunits called nucleotides. Nucleotides all share three main components: a nitrogenous base, a pentose sugar, and one or more phosphate groups. The carbon residues, or the carbons in the ring, are numbered 1' through 5'. The prime distinguishes these residues from those in the base. Those in the base will be numbered without using a prime notation. The base of a nucleic acid will be attached to the ribose's 1' position, and the phosphate will be attached to the 5' position. When a polynucleotide forms, the incoming nucleotide's 5' phosphate will attach to the 3' hydroxyl group at the end of the growing chain. There are two types of pentose sugars found in nucleic acids. The first is deoxyribose, which is found in DNA. And the second is ribose, which is found in RNA. The difference between the two sugars is the presence of the hydroxyl group on the ribose's second carbon and the hydrogen on the deoxyribose's second carbon. As I mentioned earlier, there are 4 nitrogenous bases for DNA and 4 nitrogenous bases for RNA. Both DNA and RNA share 3 of the nitrogenous bases, cytosine, adenine, and guanine. However, DNA has thymine and RNA has uracil. All of them are organic molecules that contain carbon and nitrogen. However, we classify them according to 2 groups. Adenine and guanine are known as purines. The purine's primary structure is a 2-carbon nitrogen ring. Cytosine, thymine, and uracil are what we call pyrimidines. Pyrimidines have a single carbon nitrogen ring as their primary structure. Nucleotides are capable of forming long chains with the use of phosphodiester bonds. Phosphodiester bonds form when the phosphate residue attaches to the hydroxyl group of the 5' carbon of one sugar and the hydroxyl group of the 3' carbon of the sugar of the next nucleotide. As you can tell from the image, the phosphodiester bond is right here between the 5' carbon and the 3' carbon of another nucleotide. These long chains of nucleotides form one strand of DNA. The DNA has a double helix structure. A DNA helix will have two strands that run in opposite directions, meaning that the 5' carbon end of one strand will face the 3' carbon end of its matching strand. This is what we call antiparallel orientation. In the DNA double helix structure, the sugar and phosphate lie on the outside of the helix, which forms the DNA's backbone. The nitrogenous bases are then stacked in the interior like a pair of staircase steps. Hydrogen bonds bind the pairs to each other. Now, only certain nitrogenous bases can pair or bond with other nitrogenous bases. These pairs are composed of a purine and a pyrimidine. In DNA, adenine will always bind to thymine, and guanine will always bind to cytosine. However, remember there's a little bit of a difference between DNA and RNA. So in RNA, adenine will bind to uracil. There is no thymine in RNA strands. So the easiest way to tell the difference between a strand of DNA and RNA is to locate whether or not there is a thymine or a uracil in the strand. Now, since our DNA strands are arranged in an antiparallel fashion, meaning they run opposite of one another, the strands will always be complementary to one another, just running opposite directions. Now, if somebody gave you one strand of DNA, say this strand right here, it says there are 2 adenines, 2 thymines, 2 guanines, and 2 cytosines. You would know that the complementary has to be the opposite of the nitrogenous base pairs. So adenine would bind to thymine, so you would have 2 thymine, followed by 2 adenine that binds to the original thymine, followed by cytosine binding to guanine and guanine binding to cytosine. Now this would be the same type of strand. Oops. Let me fix this. This would be the same type of strand except for RNA. Now that I've fixed it, there are adenine and uracil pairing, guanine and cytosine pairing. All right, guys, that concludes Chapter 3, Biomolecules. Hope y'all have a great rest of your day.